I don't want that to be the first thing that is heard on the recording. Welcome. All right, so the plasma membrane structure is super important because it determines a lot of cell function. This is a picture from your book that shows a portion of the plasma membrane. So if we're looking at a cell, you can draw a simple cell that looks like this. We are built out of eukaryotic cells, so our cells have a nucleus. And this would be the plasma membrane. And this picture is representing just a small portion of that membrane. Okay, so it's giving us some detailed structures. What it's showing us is that the plasma membrane basic structure is composed of little molecules, which are called phospholipids. So the golden parts of this picture are representing these special molecules, which are called phospholipids. These big structures are proteins. So proteins have a lot of different jobs in the plasma membrane, as we will talk about. So our basic structure of the plasma membrane is that it's built out of a bilayer, a double layer of the phospholipids with the proteins that are embedded. And then do you see these little blue things? That's cholesterol. So cholesterol is a structural component of all of our plasma membranes. So how many of you have heard of cholesterol, right? Because it's associated with cardiovascular disease, right? But cholesterol is necessary. It is an ingredient in all animal cells. Okay, so it helps to provide structure and stability. So we talked about 3 different parts: phospholipids, proteins, and cholesterol. So the phospholipids, a little bit more about that. In the plasma membrane, it's often described as a phospholipid bilayer. What does that mean? It means that there are like a sandwich of phospholipid molecules. So each little molecule looks like that. It's like a little ball with 2 tails. So in the plasma membrane, these are arranged in a double layer like that. So the reason they arrange themselves in this way is because the 2 little tails are fatty acids. They are lipids, they're fatty acids. Now, do lipids or fats, do they mix well with water? No. So the fatty acid tails are attracted to one another and they repel water. And in contrast, these little heads really like water. They interact easily with water. So what this does effectively is it separates 2 watery components. I'll use our little initials here. So this is standing for what? Extracellular fluid, and ICF is intracellular fluid. So the plasma membrane effectively separates these 2 watery compartments and means that you can have an environment inside the cell that is different than the environment outside the cell. And so the environment inside the cell enables you to carry out all of the processes that are necessary for life, for living organisms. Okay, so these guys are super important to us, these phospholipid molecules.
The inside of the cell being protected from It's environment, I'm forgetting the term for that.
Do you know a term that represents the inside being protected from the outside? Homeostasis? No, you stumped the professor and that means that you get points deducted. No, I'm sure there is.
Is cell wall synonymous with the plasma membrane?
Is a cell wall synonymous with the membrane? Is that your question? That's an excellent question. And the answer is no. Because animal cells do not have a cell wall. So cell walls are found in plant cells. That's what gives them the rigidity, right? Animal cells do not have a cell wall. Great question. The plant cells have a plasma membrane just like ours. It's made out of a double layer of phospholipids, but it's inside the wall. So they have a double thing going on. Yeah. Different. So this has a specific term. So the heads of the phospholipid molecules are called hydrophilic. And the tails are hydrophobic. So phobic means fearing or hating, right? So and philic means to really like something. So the heads really like water, the tails do not like water. So hydro refers to water. So that means that the little fatty acid tails repel the water, the heads love the water. So it makes this groovy barrier between the two watery compartments. It also really determines a lot about what can get into and out of the cell. Water-soluble molecules can't cross the membrane very easily. Okay? So because of the fatty acid tails. Lipid-soluble molecules can cross if they are small. So fat-soluble molecules can cross. And again, the magic of the phospholipid bilayer of this membrane is that it separates the internal and external environments. It is absolutely part of homeostasis that this is happening. So homeostasis is the ability of the body to maintain an internal environment that is conducive for cellular function, right? And so having this compartment be able to have the materials that are necessary for living processes, like producing ATP. You can't do that in the extracellular environment. So we compartmentalize these areas, and that is required for life. Absolutely. All right. So let's talk about then. So what do the phospholipid molecules do? They provide the basic structure. Okay. The proteins provide a lot of the function of the plasma membrane. So proteins, here's another picture that depicts proteins. Proteins can pass all the way through the membrane from from the external side to the internal side. Those are called integral or transmembrane proteins. Proteins can be peripheral, that means they can be attached to the intracellular or extracellular side. This little piece right here, I think it's back here as well. These to me look, god dang it, it's this time of day. These to me look like red vines, you know, licorice. But it's really a specific type of protein that is peripherally attached that looks like ropes. It is called actin. That is the type of protein. It anchors the plasma membrane onto the cytoplasm. Okay, that's a peripheral protein. And the number of phospholipid molecules is much greater than the number of protein molecules, but protein molecules are much bigger. And so the membrane is at least 50% proteins if you just look at mass or weight. Okay, so proteins are important for the function. Let's talk about why. So these are some different kinds of proteins that might be found in the membrane of cells and some jobs that they do. The proteins act as receptors for chemical messengers. That's one job that they might have. So this is depicting an integral protein, one that is embedded in the membrane, and it has a very specific shape that can attach to a chemical messenger that is on the outside in the extracellular fluid. Chemical messengers tell the cell what to do. So a chemical messenger or chemical signal, as we sometimes call them, the most common chemical messengers are neurotransmitters and hormones. So neurotransmitters are produced by neurons. Neurons are also called nerve cells. So the way that nerve cells tell other cells in the body what to do is by sending a messenger, which is a neurotransmitter. Right? So an example is that every time that you move, you produce neurotransmitters that attach to skeletal muscle cells. Okay, so there are proteins in skeletal muscle cells that respond to a neurotransmitter that says it's time for you to move. Okay, so that's an example. Another example are most hormones act in this way. They are released into the blood, and when they travel to a cell, the cell has a receptor that is a protein in the membrane. And the receptor says, okay, I see you, hormone, and binds to it, kind of like a baseball mitt, it catches it. And then that changes the cellular function. Let me give you an example. You have heard of adrenaline. Have you heard of adrenaline? Another name for adrenaline, the more technical name is epinephrine. Epinephrine is a hormone that is produced in the adrenal glands. When you produce adrenaline or epinephrine, how many you're stressed? When you're stressed. How many of you have gone to the boardwalk at Santa Cruz? I hope so. Should be a field trip. Because we could experience stress. Instead of doing the exam, we could just have it. And so like the giant difference for that. You know how you feel when you come out of the dark and you just start to do that drop and your stomach, oh my God, I can't cope. It's worse than anything. What's happening, of course, is that you know that what happens when you get stressed or afraid, you know the axe murderer is right behind you. What happens to your body?
Fight or flight.
Yeah, it's fight or flight. It is. And you experience that increased heart rate. Your pupils dilate. You feel mentally alert, right? You're going to be so alert during the exam. It's going to be fun and exciting. So that adrenaline is released into the blood and it interacts with a lot of cells in the body to produce those responses. So that's one job of proteins. You'll notice that this is depicting a protein that is right next door that is taking whatever the messenger is and breaking it down. You don't want to be in a fight or flight situation for hours and hours and hours. So once the messenger stimulates a response in the cell, this protein is going to break it down. So it acts as an enzyme. So an enzyme is a type of protein that can break apart another molecule or build it up. But in this case, it breaks it apart. Another function of the proteins in the membrane is that they can act as channels. Because remember, I said that water-soluble molecules or particles cannot cross into the cell because of the fatty acid tails. They repel things that dissolve in water. So if you're water soluble and need to get across, you can have a protein that acts as a channel. Here's an example of a substance, a solute, something that dissolves in water. A solute can cross, and the example I'm going to use is glucose. What is glucose? Sugar. Okay. It's our blood sugar, right? Why would you need glucose to cross into cells, do you think? Do you have any ideas?
Body, the brain especially.
Absolutely, you need it to run your cells, especially your brain needs a lot of glucose because your brain uses a lot of ATP or energy, right? So glucose is the raw material for producing the energy that your cells need to function. And so glucose is water soluble. It's a sugar. When you put sugar in water or coffee or whatever, doesn't it dissolve? Well, it dissolves in water, and so therefore these guys aren't going to let it cross. Okay, so you need a channel to let it in. That's pretty important, isn't it? So that happens in every cell of your body. Some channels are closed most of the time, and they open only under certain circumstances. Some channels open because of electrical current. You don't understand probably, or maybe you haven't, I'm sure you understand, but you haven't thought about it maybe, how darned electrical we are. We run on electricity. And our electricity, instead of being electrons in an electrical cord like this, our electricity is sodium and potassium, and those little electrically charged particles that run our neurons and muscle cells. It's incredible.
Are there proteins that are constantly open?
Yes. There are proteins that are constantly open. So there are protein channels that are always open. There are other channels that only open if they receive the appropriate signal or if they are electrically stimulated, for example. Well, for example, there are in your neurons, there are channels for sodium. Sodium is really important in our body. That's the chemical notation for sodium, Na+. That's something you're going to have to know. And sodium is, do you notice the plus is because it's electrically charged? So the example is that if there is a stimulus to a neuron, it will produce an electrical signal that opens the channels for sodium, and sodium will rush in. And that produces a wave of electrical activity that enables you to do whatever you're doing. Short answer to a complicated situation. We're going to go through that in huge detail. not too much detail, but just the right amount of detail. So I can't wait. You make me excited to talk about neurons. So some proteins, do you see this little green thread? That is a chain of carbohydrates, which are like sugar molecules. Your whole plasma membrane is coated with sugar. It's got a sugar coating like an M&M. And that helps to protect it from toxins and things but sometimes, like this is a protein that has this carbohydrate chain attached. This is called a cell identity marker. You know, like one of the problems with organ transplants is that when somebody receives an organ transplant, they reject it. That's because your cells have unique markers on them that say, I am myself, I am who I am. And it also identifies anything that goes into your body as being either self or non-self. So if you encounter something foreign, this will say, you're not part of me, and I'm not going to handle it. So that is a cell identity marker. This is also a signal to other cells, don't attack me, because I'm part of you, dude. So like if your white blood cells are running around saying, I'm going to attack anything foreign, you don't want them eating up your own cells. So your cells have proteins with markers that say, me and me. And that is how that works. And then the other thing is that many cells are tightly woven together. For example, your cardiac muscle cells, your heart muscle cells, your epithelial tissues, the cells are tight. They do not separate. The way that they are joined is that there are proteins in each of the membranes that glue them together. So those are called cell adhesion molecules, So let's review quickly. Functions of proteins in the membrane. Receptors for cell signals or chemical messengers. Enzymes that break those messengers apart. Channels for water-soluble substances. Some of them are gated, some of them aren't, or closed. We call that gated. Cell identity markers and cell adhesion molecules. Yes.
You mentioned toxins earlier, what effects does the membrane experience from experienced toxins? I mean, there's so much like out there, like glyphosate, whatever chemicals and heavy metals that get trapped in our body, never leave. And I was doing some research that it has a lot to do with the membrane. So how does exterior factors affect the cell membrane that they never leave the body?
That is a really good question. And I'm going to answer this in two ways. First of all, in the extracellular fluid, there are always chemicals, even if we're not talking about external environmental toxins. There are chemicals that could damage the cell. So this carbohydrate little shell, I'll call it, protects against our natural kind of chemicals. The kinds of damage that can cause, as well as external things like you're talking about metals or whatever we might take in from the environment, they can rip the cell membrane apart. This plasma membrane is fairly fragile, and so it can be torn apart by environmental assault. If you do that, then the cell will die. So, and remember, there are different levels. So environmental toxins sometimes don't impact a specific cellular membrane. They might impact what are also called membranes, which are sheets of epithelial cells that line, for example, the respiratory and digestive tract. So that almost is a different biological level. So it's interesting. Yeah, a great question, a very complicated question. Okay, so the point is that membrane proteins are important. Would you agree? Would you say that proteins in the membrane are important? And could you list some reasons why? Okay, good. All right. So this is just a summary of what we just talked about, so you can read that on your own. All right, the membrane also is important because it allows material into and out of the cell. That process is called movement of materials across the membrane. All right, so movement of materials across the membrane can be either passive, the processes can be passive or active. Passive means that it does not require cellular energy in the form of ATP. Passive does not require energy from the cell. Active does require energy. So the passive types of movement are diffusion and osmosis. Active types of movement are active transport, where you use proteins that actually change shape to move material across, and special processes where the cell engulfs material. The only one I'm gonna make an executive decision and say that the only one that I really care that you know about is phagocytosis. So you can cross out pinocytosis. So phagocytosis is literally cell eating, where a cell engulfs material and brings it into itself. Exocytosis is where this is the opposite, where a cell creates a little package inside itself and exports that package. Okay, so let's talk a little bit about these and why they're important to us. Okay, so diffusion is literally, the definition of diffusion is movement of molecules or particles. It doesn't have to be molecules, it can be particles from areas of high concentration to areas of low concentration. This happens outside our body too. This is not just confined to biological processes. If you take a bottle of perfume and squirt it, the molecules will diffuse. Areas where it's highly concentrated to areas where it's less concentrated. You put, I don't know, a tablet of red dye in a glass of water. What happens to the water? It eventually, if the tablet dissolves, doesn't the water change color? That's because the dye diffuses through the water. So areas of lots to areas where there's not very much movement, right? So some examples of why this might be important is lipid soluble. Lipid soluble means something that dissolves in fat, can float right through the plasma membrane if they're small. An example of fat soluble particles that come through, or molecules, are steroid hormones. So do you know what steroid hormones are? Steroid hormones are hormones that are small lipids that include things like estrogen, testosterone. So they are very small. They're little pieces of lipid that can float right through. So they diffuse from areas of high concentration to areas of low concentration. Water soluble, as we mentioned, like with glucose, have to go through a channel. All right. So other examples, I'm gonna just give you another example of diffusion. Particles that can diffuse across the membrane include gases like oxygen. So oxygen is O2. So the fact that O2 is very small relates to a lot of the structure of our lungs. So the little tiny air sacs in the lungs that you ultimately breathe the air into provide an enormous surface area for diffusion of oxygen. So oxygen moves easily across the plasma membrane. It diffuses from areas of high concentration to areas of lower concentration, for example, and then you can transport it throughout the body in the blood. Okay, diffusion is very important to us. Yeah,
I know like breathing is a very fast process, and when you inhale, your oxygen goes into your cells and then takes it throughout your body, and then you exhale, it like exhales the CO2.
exactly.
Like, I don't know, it seems sort of mind blowing to me that the air that just went in is coming out different air, like, is it a very fast process?
It is very fast. So the question was, when you breathe in, you breathe in a mixture of gases, but the oxygen is what's important. So it diffuses into the blood, and then you breathe out carbon dioxide. What's up with that, right? So I'm not going to answer the question. And the reason is because we're gonna go into that in a lot of detail a little bit later. But I'm so delighted that you asked it, and if both of them have to do with diffusion, I will say that. So, but that's a cool question. Save it, we're gonna save it. All right. Osmosis is about water, the movement of water. And one of the things, how much water do you have in your body? I mean, how much of your mass, I should say, or how much of your weight is composed of water? How many percent? I heard 80. Okay, I heard 30. 60. We'll take 60. 60 to 70 is typical. So 60 to 70% of your mass is water. It's very important that we maintain water balance, and we keep water balanced on opposite sides of compartments. Like we have to balance the water inside the cell in the intracellular fluid compared to the extracellular fluid. Can't have more on one side than you have on the other. Or else, for example, if you have too much water in the cell, it will burst. Okay, so water balance is what we call that. And that's very important, and that relates to osmosis. So osmosis is the diffusion of water across a selectively permeable membrane. Now, that's a mouthful. It just means that it's the movement of water across a membrane that allows some things in and not others. A selectively permeable means that you let stuff in. Okay, so selectively permeable means that you only let some stuff in. So water can, here's the sitch with our cells. Water can move across the membrane, but solutes, things that are dissolved in the fluid on either side, typically cannot, because most of these solutes are water soluble. So they're repelled by the plasma membrane, by the fatty acid tails of the phospholipids. So you can move water back and forth across, but you cannot move solutes, typically. Okay. So that's what I put up here. The membrane is more permeable to water than it is to solutes. So first question is, why can water cross and not these guys? Because water is actually, well, it's water soluble. It's what we would call polar. Most polar substances can't cross, but remember, what is the chemical notation for water? How big is the molecule? one hydrogen, no, two hydrogen and one oxygen. How many atoms is that? Is that a big molecule? It's teeny, 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 tiny. So one way it can move across is that it can slip through the phospholipid fatty tails. But the other way that it can move across is that there are in most cells, there are channels for water that are called aquaporins that are often just open. Okay, so water can move back and forth across. So that was only relatively recently discovered. I know 1990 is a long time ago, but in the grand scheme of things. So you have to maintain a net flow of water. So you have to maintain it in balance. This is actually a microscope slide picture of red blood cells. And you can see that this cell is normally shaped. So this is a normal shaped red blood cell that has a round, beautiful structure. These are spiky. Do you see that? Why are these spiky? Because this person was given a solution, perhaps like an IV solution, that would have had a lot of maybe sodium in it. And let's think about that. If we put our red blood cell, so your red blood cell normally has a specific amount of solutes inside. There's a concentration of solutes, which is actually 0.09% in a normal cell. Let's say you go to the emergency room and somebody accidentally administers a sodium solution, sodium chloride, let's say, solution of 5%. So it's 5% concentrated with solutes. Which of these has more, the cell or the IV solution? Which has more solutes? Which has a higher solute concentration? It's 5%, so this is 5.0. So this is like $5 and this is like 9 cents. So this one is more highly concentrated. Do you agree? What's going to happen to the patient? So you've got a ton of sodium in it. That's a notation for sodium chloride. So you've got just a lot. Well, I will tell you what happens. Solutes suck. And you should memorize it. Because the solutes, the water moves toward the solutes. So water will move outside of the cells to the higher concentration of solutes. So water diffuses from areas of high concentration of water to lower concentration of. So there's relatively more water here because there are fewer solutes. Does that make sense? And then there are more solutes out here and relatively lower levels of water. I think you can imagine this. If you put a really, if you have a really densely concentrated, like, I don't know, salt water, the water will move toward the solutes to make it even wetter. I don't know if that's a good example or not, but this is what happens in our body. So we maintain our solute concentration in equilibrium, in equal balance, in the intracellular fluid and the extracellular fluid. Otherwise, your cells would burst or shrivel. Okay. So osmosis We have mechanisms to maintain that. We maintain very specific levels of solutes in our blood. I know it's a little bit complicated, but that's how it works. Yeah.
So the spikiness on the cell, is that representing the water leaving the cell?
Yes.
That's literally a
It's-
visualization of.
yeah,
the
it's-
cell,
It's gonna shrink.
and then it will shrivel after that?
Yep, it will. So it's actually starting to shrivel. And yeah, you can see that under the microscope. Yeah.
But the soluble cell into the cell.
That is such an important point. The solutes cannot move into the cell. So it's the water that moves to create the balance between the solutes on the inside and the solutes on the outside. So water can move, the solutes typically can't. Really good point. Yeah. This is, some further, just trying to explain this a bit more. This is looking at a red blood cell, which is often the cell that is affected by this. So this is the fluid in the blood, right? It's got solutes in it. In a normal situation, you would have the same concentration of solutes inside the cell as outside. And there would be a net movement of water. Water's moving in and out, but no more is leaving than is coming in. And that maintains a normal blood shape. Let's say you have a solution where instead of a 5% sodium solution, you do a 0% concentration of solutes. Then you're gonna dump that into your blood. And what will happen is that the solute concentration inside the cell is greater. Water will move in, and what will happen to the cell? Eventually, it'll get bigger, and then it will, if enough water comes in, it will burst. This is the situation that I drew up here. If you put in a highly concentrated solution around that cell, the solutes will suck the water out and the cell will shrivel. Okay. We have names for this. In the normal situation where the concentration of solutes is the same on the inside and the outside, we call the solution an isotonic solution that will not change cell shape. If the solution has fewer particles, it is called hypotonic and water will move into the cell. If the solution has more concentrated solutes, it is called hypertonic. This is really important clinically for that reason that I talked about. Often electrolyte balances lead to water imbalances. And it can really cause a lot of problems for the individual.
So does this relate to like extension and like blood pressure? Or not like huge salt?
Not typically, but it's an excellent question. Definitely water balance does. So, but not the solute concentration. So, good question. That's another thing we'll come back to. Yes. Explode, the person dies. Yeah. Pretty much. Yeah. Yeah, you can kill somebody. Yeah, so don't do it. So that's why, that's why you need to know this. I know I should just say that outright and be transparent. I want you, if I come into the emergency room, I don't want you to make a mistake if you're a nurse in there. So, and the reason is this, you can kill somebody. So, I mean, I don't want to scare you, but it's true. This matters. So, yeah. All right, so active transport requires ATP. It often requires proteins that act as ferries or transporters across the membrane. That's all I want you to know, really, except that this is the main example that I want you to know. So the main example of active transport is the sodium potassium pump. Okay, so this is the sodium potassium pump. That's a weird term, but it really is referring to these specific channels that maintain the balance, the appropriate balance of sodium and potassium on either side of the membrane. So they actively transport sodium out and potassium in. So the notation for potassium is a K positive. K positive stands for potassium, Na plus stands for sodium. Now, the reason this requires ATP is because normally there is more sodium outside the cell than inside the cell. So normally sodium is in greater concentration outside the cell than inside. So what would be the natural tendency of sodium to move? What direction would it normally move? If there's more out here and less in here, how would it want to? It would want to go inside the cell because you want to go from areas where there's a lot to areas where there's a little. But the cell is saying, no, I don't want that. I need to have more sodium on the outside than on the inside. So it is going to take energy, ATP, and push that sodium out so that you maintain a lot of sodium outside. And you do the same with potassium. Potassium is more highly concentrated inside. The natural tendency would be to move out. The cell is saying, no, I need this imbalance between sodium on the inside and the outside, potassium on the outside and the inside. Okay, so it's taking energy to create an imbalance or disequilibrium. Why do you need them? Because it produces an electrical charge that you can use, you exploit that difference, especially between sodium on the inside and the outside. It creates electricity that you can harness for either passing nerve signals or producing movement. So you need the electrical charge, you need that imbalance of those molecules to both fuel or to power your nervous system and your muscular system. Let me just tell you that this process of the sodium potassium pump throughout your body, this accounts for 50% of the calories that you eat every day. I want to say that again. This is so important to maintain this disequilibrium across the membrane that about half of the calories that you eat every day are used to produce enough energy to power all of these proteins that act as sodium potassium pumps. So you require ATP to move sodium out and move potassium in. Why do you need to do that? Because it's an electrical charge across the membrane that you use for your nervous system and your movement, in addition to other things. Okay. Is your mind blown? Yeah, I understand. All right, let's finish this up. Don't we have 5 more minutes? 4 more minutes? We have 4 minutes. So endocytosis, the only one that I want you to know is phagocytosis, which is cell eating. And one important example of why this is essential for us is this is looking at the process. So this is a cell that is literally changing its shape to engulf something from the outside. This particular cell is a white blood cell. So white blood cells are very critical to keep you well. They help protect you from diseases, especially infectious diseases. So what this white blood cell is doing is it has recognized that there are bacteria that have invaded you. And it is saying, wow, this is what I'm good at. So it's gonna eat those little son of a guns up. Thank you. And it will engulf them, embrace them, bring them into themselves, and it will digest them. Isn't that amazing? If you are invaded with bacteria, you have white blood cells that can engulf hundreds of bacteria at once and get rid of them, so that you may not even experience symptoms of being sick. Okay, so that is phagocytosis, eating, and that's where we'll stop. Okay. So, recording.